We will do it maybe short. We are half. So, did you read the paper?
Yeah.
Okay. So the last thing we need the Windows B. So the last thing we saw was this thing. When we treated the cells with this global methylation inhibitor, many genes changed, and our results indicated that circadian clock was strongly affected by this drug, as we can see from the previous data about the period, right? But when we looked at separately at the genes that are induced and the genes that are inhibited by DAA, the genes that are induced, they were all of them involved in RNA processing. And the genes that were reduced, you can see that 3 out of the 4, it was about translation regulation. Regulation of translation. You see, translational elongation, viral transcription, which is, it's a little bit strange to explain, but viral is also RNA, right? So some of the protein have also been involved in how viral RNA is translated into the cell. So it's a little bit strange term, but it's also mean translation, this one. And then here it's also about translation. So everything is about RNA. All the genes that were found to be strongly affected by DAA were genes that were involved in the processing of RNA. So this kind of told us that maybe what we are doing with DAA is affecting how RNA is processed. And therefore, so what's the next thing? What's the next thing? So we know maybe from, if we go to the discussion, I want to find this paragraph for you. So yeah, this is the one I want to go over because I explain what we got. So I explained in the paper, I explained this result a bit more carefully to convince the reviewers, and that this one here. We then separately analyzed the 4,360 induced and the 5,120 repressed genes against the 9,480 significantly regulated genes as background. So whatever this is analysis, we wanted to do this one, right? The analysis I showed you before. And then the most significant match, RNA processing, is described as any process involved in the conversion of one or more primary RNA transcripts into one or more major RNA molecules. Do you understand this? Yeah?
Capping and
capping, splicing, Among genes, also, what you do not see from the figure is what are the genes that are induced. So here I explain some of the interesting genes that were induced by DAA. Among genes induced by DAA, our dataset included in this GO means gene ontology, function of genes, are notably the RNA M7G cap methylase, RNMT, and RNMTL1. 1L means like. So this is like this one. We don't know what is the function of this one, but this is a protein that actually methylates the cap of messenger RNA. As well as the M2O ribose cap methyltransferase, the M6A demethylase LKBH5, other RNA methylase such as MTTL2A, MTTL2B, and RRP8, adenosine deaminases such as ADAR and ADAR1, many other RNA binding proteins such as NCBP2 and RBMS1, splicing factors, blah, 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 all to do with RNA processing, methylation, and maybe the very interesting one we thought about was like, why I say this LKPH5, because it's all to do with methylation. The strongest genes that were regulated by DAA were genes that have something to do with RNA methylation. Somehow, when you treat the cells with DAA, which is a methylation inhibitor, all these genes that are making methylation increase. Right? Hello. Don't worry. So that was kind of a nice indication, is that when we use this methylation inhibitor on the cells and we take a look at the genes that are induced, the genes that are induced the most are the genes whose function is to methylate RNA. Sorry. It's the fourth lecture today, so I'm sorry if I'm telling you rubbish. Okay. So all these genes have something to do with RNA methylation. So to us, this is it's an indication that maybe what we are doing is that we are inhibiting RNA methylation, and the cell in reaction is inducing all these genes that methylate RNA. So it tries to compensate for the inhibition we are doing. This is what we thought it was happening. Okay. So this is why we decided to look at RNA methylation. But if you remember the discussion we had last week, it's not only that. Not only the genes that responded to DAA were about RNA methylation and related processes, but said, supporting this hypothesis, RNA methyltransferase are known to be the most sensitive to the decrease of the methylation potential. Carmel and Jacobson in 2001. In particular, DAA is a strong inhibitor of the internal M6 methyladenosine. Backlund, 1986. So this is the last piece of information that we included in the paper, because it has been shown before that there are many RNA methylases, but the one that is the most strongly inhibited by DAA is this internal N6 methyladenosine. Okay. All right. So now we need to explain about things. What do you know about RNA methylation? I'm asking you, what do you know about RNA methylation? What do you know about methylation? Yeah, DNA methylation. We have heard about DNA methylation. What else? Yeah. So it does really not really protect. If anything, it accelerates the processing of RNA. It really doesn't really protect. Some of them protect. It depends which methylation. But the point is that before I did this research, I had no idea about RNA methylation. The methylation that most people know, you can ask around, is, well, at least 3 years ago, is histone methylation, DNA methylation. But RNA methylation, people usually don't know, because you have very rarely in textbooks they talk about RNA methylation. But actually, RNA methylation, there is a lot of it. The thing that most people know, it's the methylation of ribosomal RNA and tRNA. These are the most well-known methylation. And the methylation of ribosomal RNA, as you said, the methylation of ribosomal RNA is important for the structure of the ribosome. It will make the structure more stable. Okay? And the same is true for transfer RNA. Transfer RNA are also methylated, and it's important for the structure of transfer RNA. But ribosomal RNA and transfer RNA, they have a specific structure, right? They need to work. But messenger RNA is just It's just a string coding for protein, right? But it's also methylated. The problem is the function of methylation is not known. So maybe 40 years ago, people showed that RNA, that messenger RNA was methylated, but the function is completely unknown. At that time, it was completely unknown. We know it's there, but what it does, we don't know. So, I can show you a talk which I gave some time ago about RNA methylation, and it's a slide that shows this one. So what you have here is as you can see, this is the 5' end of the messenger RNA. Okay, not the polyadenylated end, but the 5' end. And this is the first transcribed nucleotide. Do you understand when I say the first transcribed nucleotide? This nucleotide had its complementary on DNA. Okay, so this is the first transcribed nucleotide. This is the second and the third. But in addition, the 5' end of messenger RNA has a guanosine. It's called the cap. Okay, but you can see that how this guanosine is linked to the first transcribed nucleotide is completely different from the normal link. This is the normal link that we all know. But you can see there is a phosphate, there is a triphosphate bridge. This guanosine, you cannot find it on DNA. It's added after. Once this has been synthesized, maybe 20 nucleotides have been synthesized, then this guanosine is added onto the end. And the role of this guanosine is to protect the 5' end of the RNA. But it's not all. It's not all. Because, as you can see here, there is a methyl. This is not guanosine. There is a methyl here, which does not belong to guanosine. So this guanosine can be methylated. Not always. It can be methylated. In addition, the first transcribed nucleotide can be methylated at the ribols. And the second transcribed nucleotide can also be methylated at the ribols. The function of this methylation here and here is not known. We know it's there, but we don't know the function. And this nitrogenous base methylation of the first transcribed nucleotide can also happen. And you can see they have very strange names. Like this 7-methylguanosine, this is the name of this methylation. But this is the symbol, M7G 5' PPP 5', X is this one. Okay? It can be anything. But if X is A, then this can also be methylated. This is why you have M7G 5' PPP M6A, this one here. Okay. So they have a lot of methylations. 1, 2, 3, 4 methylation in the cap. And we don't, this one we know more or less what is the function, but these ones we don't know. And it's sequence specific. But in addition, there is another one, which is an internal methylation. When I say internal methylation, it is inside the messenger RNA. Not on the cap, but inside the sequence. Exon or intron, whatever. Inside the sequence. But not only it is inside, but it is sequence specific. It needs to be on an adenosine that is located in this kind of sequence. You know what this is? It's called the matrix, and it shows what is the most likely sequence to be methylated. And as you can see, the most likely sequence is GGACT. And when you have a sequence like this in messenger RNA, the adenosine is usually methylated. But you can see that it can also be AACT, AAACT, or AAACA. So there are some variations. But when you have this sequence, it can be methylated. And even more complicated, sometimes this sequence exists, but it's not methylated. So we still don't really know why some of these sequences are methylated and why some of these sequences are not methylated. So this is what we knew about at that time. Okay? This is what we knew at that time. So to go back on the paper, what available information we had before we published this paper was that among all this methylation in messenger RNA, it was known that DNA is a strong inhibitor of the internal M6 methyladenosine methylation. Then, since the circadian clock involves a transcription feedback loop, period elongation via RNA methylation-dependent RNA processing mechanisms would be a thought-provoking and novel possibility. So we explained why we then studied M6A methylation, RNA methylation. All right. So what's the next step? What kind of stuff would you do? We sort of say maybe what we have is its elongation of the circadian period by inhibition of M6A mRNA methylation. So what would you do next? What would you do next?
To invest which kind of mRNA is affected by the inhibition of the M6 methylation.
Yes, you could check which RNA are affected before that.
Check which RNA are methylated at that side.
Yes, you can check which RNA are methylated. You can do that. Before that, what is one of the first things you can do? What we have here is a suggestion. Okay, we have not proven anything yet. We have only suggested, our data suggests that M6A methylation is inhibited by DAA. Prove that it is. We need to show that it is. And in the same time, if we can show that it is inhibited, we, in the same time, maybe can show that, yes, it is inhibited, but which M6A? Because M6A can be on mRNA or not. So if it is inhibited, which transcripts have their M6A inhibited? So this is what we did. And I think you will understand this one because of what we saw previously in this lecture series. Do you remember CHIP analysis? Chromatin immunoprecipitation. The paper we read before. How did they quantify the time-dependent binding of clock and BMAP on DNA? Do you remember how they did it? How did they do it?
Define the protein
Yes.
antigen to the beast, and then after that, they take a large amount of cell and let the antigen connect to the proteins.
The antibody, not the antigen.
The antibodies connect to the proteins, and then they digest all the place DNA that is not bound by the protein.
Yeah, that's it. So using an antibody that is against clock, for example, you can immunoprecipitate clock with its target DNA, and then you can sequence the target DNA and how much there is, and this gives you an idea of the binding. Now, what we are doing here is not DNA, it's RNA. Okay? And using a similar method, we also want to quantify not only how much methylation there is, but where it is. How could we do? How could we do that? It's not DNA, it's RNA. How could we do that?
Do you also use the microarray and the probes?
Not microarray, not microarray.
Use the anti-M6 antibody.
The M6A?
The antibody that binds to the it is methylated.
Yes. So you get an antibody that is against methylated adenosine. That is the same adenosine that is methylated in RNA. So it's just an antibody that recognizes M6A, okay, the methylated adenosine. Then what? You have an antibody that has been raised against M6A. By the way, M6A, so that you know what M6A means, M6A is this one. This one here. This is adenosine, right? This is adenosine and this is M6A. It's the same position that is methylated here, okay? It's the same position. So, we have an antibody. What do we do with it?
Instead of digesting the mRNA, we sequence it and finding out and review which mRNA it is.
So what the first step do you do? So you have your cells, and it's an experiment in which we want to show that deoxyribonucleic really inhibits M6A. So the first thing to do is that you have one dish with cells treated with nothing, with control, and one population of cells that is treated with DAA. What is the first thing you do? Step 1. Extract RNA. Step 2.
Isolate the mRNA.
Huh?
Isolate the mRNA.
Yes, that's the step 1. Isolate mRNA. Isolate RNA. That's the step 1. Be careful of my word. Isolate RNA. That's the first step. What is the second step?
The RNA precipitation method.
That's the third one. Actually, that's not the third one. That's the fourth one. Before. Remember what I told you earlier today. Not this lecture. Not you. Not like Kagakuigo, no. I told you that ribosomal RNA and tRNA also methylated. Now, you have to know that 98% of RNA in the cell is ribosomal plus tRNA. So if you purify RNA from the cell, mostly you get ribosomes. You really don't want ribosomes because of the... Yeah. So first step, you isolate RNA. Second step, you isolate messenger RNA. then what? Remember, what you said is fourth. The third step is important for the location of the M6A. It's important to know the location. If you directly go to the fourth step, you will not know where it is. Not precisely. So the third step is very important.
Sequencer?
No. That's the fifth one. I like these games. It's really the same as this chip thing, as this clock analysis or RNA polymerase II analysis. That's very similar. That's very similar. So extract RNA, extract messenger RNA. That one. Okay, let's just pretend that I'm not always doing this third step. I'm not always doing it, like I did it not so long ago, and I didn't do the third step, because it depends what you want to do. Let's skip the third step, okay? So you purify RNA, messenger RNA. Using the antibody, you immunoprecipitate your RNA, and then you sequence. You will know that maybe RNA A is methylated. And RNA B, because it's very low amount in your immunoprecipitated sample, it's not methylated. Okay? So you will only know which RNA is methylated, but you will not know where it is methylated. You want to know where, because one RNA can be very long. It can be 3 kilobase, it can be 4 kilobase, it can be even longer than that. You want to know where it is. Is it methylated in an exon? Is it methylated in the UTR? You know the UTR? UTR is, so RNA can be very big. And RNA has essentially, it depends what you say, but there is essentially 3 parts. There is the CDS, or the coding sequence, and this is what goes into protein. And you have the 3' UTR and the 5' UTR. And UTR means untranslated. Untranslated region. So it's not coding for protein, it's only regulatory. Okay? So you want to know where it is. Where is methylation? This M6A. I told you it's inside the RNA, but we don't know where. Is it here? Is it here? Or is it here? We want to know that. So how could we do? The step 3. The mysterious step 3. It's really the same as clock chip thing.
Because the methylated place is always the GGACT and
Yeah, so by computer, by analyzing the sequence, you may be able to know where it is methylated. But the problem is, as I said earlier, sometimes you have the sequence, but it's not methylated. If you think about this, what is the likelihood of having GGACT? What is the likelihood of having this? What's the likelihood of having this one? What is the likelihood of having this sequence in your messenger RNA? No, not so low. You will have once in how many nucleotides? This. If you look at messenger RNA, you expect to see one GGACT how frequently? 1/1. It's just normal probability. Yeah, exactly. 4 to the power of 5, right? 4 times 4 times 4 times 4 times 4. But from the slides I showed you before, you can also have AA. So then it becomes even more frequent. Now, methylation sites are much less frequent than that. Sometimes you have the sequence, but it's just not methylated, and we don't know why. So just by computer, it's not enough. You really must check where each messenger RNA is methylated.
When the antibody is binding to the mRNA, can the RNA sequence at that place be sequenced?
Yes, this is what you want. Actually, what you want is to sequence only the part of the messenger RNA that has the antibody bound. So how do you do that?
Find it and then use restriction enzymes again.
That's very close. That's very close. Unfortunately, restriction enzymes, it's a DNA thing. They will break DNA at specific sites. There are no specific RNAs. Yes, so if you remember, you used DNAs or you fragmented DNA before you immunoprecipitate clock, right? You want to know what is bound. So as you said, DNAs will only degrade RNA, will only degrade DNA that is not bound to clock. Okay. So you can do a little bit similar here, but you have to do a little bit differently. Using RNAs will not work because RNAs will just degrade everything. RNAs are extremely powerful. Okay.
Could you see DNA?
Huh?
Could you transcribe it back into DNA?
You have to do that at some point. But not at that time. Because if you copy it, then you will lose methyl. So what we do first, it's not using enzymes, but we will just, by acid treatment, by chemical treatment, not enzymatic, we will break the RNA. I'm just showing break. I'm not removing sequence. Okay? I'm just breaking the RNA. I am just breaking the RNA like this. So, step 1, extract RNA. Step 2, extract messenger RNA. Step 3, fragment RNA. So what you want to do is you want to fragment your messenger RNA into maybe 200 nucleotides fragments, just by chemical treatment, using heat and maybe like a magnesium ion, typically. And then you do immunoprecipitation. That way, you will only immunoprecipitate the fragments that are methylated. And because these fragments are small, then you can just sequence them, and then you will see, aha, in my immunoprecipitated sample, I have a lot of this fragment. So this fragment has a metil. And then you can check that in this fragment, yes, indeed, the sequence GGACT is present. And then you can quantify this whole thing. And this is the next data here. This is exactly what we did. So you can have here control versus DAA, mRNA extraction, mRNA fractionation, M6A RNA IP means immunoprecipitation, and then you sequence methylated RNA. And what we observed very nicely is that if you check per 1, per 3, this is reverb alpha. It's the gene name of reverb alpha. This is another gene, DBP, you know maybe. And what we checked is only you check green, this is control, and red, this is DAA. And we can see very nicely that we have big peak. It's exactly the same as the clock thing, okay? So when you have a tall peak, it means that you have a lot of methylated RNA at that location. So we had a tall peak here in per 1. This exon, it corresponds to this exon. We had the tall peak here in the 3' UTR of per 1. For per 3, we had a big, very tall peak here in this exon here. You don't see them, you can see on the paper, but this is the gene structure here. So these thick blue lines mean the exon, and the thin blue line means the intron. So what you can see anyway, we have detected methylation sites in many of these clock genes, and when we use DAA, you can see that the size of the peak, the height of the peak decrease. So this shows that we have really inhibited M6A RNA methylation. But we want to show something else as well. This I will not explain. This is quite complex, so I will not explain this one here. If you want me to explain, you can ask a question later, but I will not explain this one. My boss didn't explain it. And okay. So this is what we get. What's the next thing? So we have shown using this method that not only some clock genes are methylated, that the messenger RNA of clock gene is methylated, but that DAA has inhibited methylation. What's next?
Find out what the effect is.
Yes, that's next, that's later. Now we need to do a kind of control that, yes, we have confirmed that RNA methylation was inhibited, that M6A was inhibited. What could we do? What could we do next?
Check destroy the rhythmic expression of any of these methylated mRNA will have an effect clock or not.
Destroy the rhythmic expression of all these methylated RNA. What data would you have? Destroy the rhythmic expression of methylated RNA.
And
What?
then see it, and then we will have which kind of rhythmic expression of mRNA will have effect on this clock.
That's an interesting thing. It's very close to what we are currently doing, but if you destroy the rhythmic expression of a methylated RNA, it will not tell you the role of the methylation. It will just tell you the role of the rhythmic expression, right? Some people have done that. Some people have, for example, they used PER2 knockout animals, and they have expressed PER2 always at the same level. So they investigate the role of PER2 without oscillations. But it's not the role of methylation, it's the role of the oscillations. The next experiment has to be a kind of control. It's a kind of control that we show, yes, many places in messenger RNA can be methylated, M6A, cap methylation, and so on and so forth. And we have shown that DAA indeed can inhibit M6A. What should we do now? We want to write a paper, as you notice, on M6A, right? We want to study M6A, so we have to sort of say, hey, M6A is important for the clock, maybe more than other methylation. So what should we do?
Inhibit the auto methylation and show that it doesn't affect the clock.
Yes, indeed. Indeed, we need to inhibit other methylation and see if it does not affect the clock. Unfortunately, we cannot, at this time, we cannot inhibit other ones. So what else we can do in the same thing? Something very close to that. Not using any other, it's possible to specifically inhibit some other methylation. That will come later. Here it's about DAA, right? We showed that DAA inhibited M6A. So what next? As was suggested by Carmel and Jacobsen and by Backlund, M6A is one of the methylation sites that is inhibited the strongest by DAA. So what should we sort of show here after this data? After showing that M6A is inhibited, what should we show? What should we show?
What M6A is and what the mRNA disease.
That? Say, come again?
So what the mRNA is translated to, which protein is translated into, what protein it affects.
Yeah, we could check. That's true. We could check to see which protein is affected. I mean, how inhibition of M6A change protein expression. We could do that. Before that, we are still in the process of convincing people that among all potential methylation, M6A explain a large part of our data. Not only among many methylation in the cell, but among many RNA methylation in the cell. And not only among all RNA methylation in the cell, but among messenger RNA methylation, the M6A is the most important to explain our data. So what should we show?
That one's present in all of those genes. That M6A is present in all of the genes which have been affected.
Yeah. So this is this one, right? This is this data right here. Is that for all the clock genes that have strong methylation peak, we have a decrease of all of them, right? So what should we show to accompany this data? Now, we should show that other messenger RNA methylation are not inhibited. We should show that M6A among messenger RNA methylation is inhibited, but the other one not so much. So this is what we did next time. Next, this is what we did. So in this case, so this is this figure here. In this figure, it's the same experiment as the previous one, but in this case, we did not fragment the messenger RNA. We took RNA, we purified the messenger RNA, and then we directly immunoprecipitated. So we knew how much each RNA is methylated. But we did not, at that time, we did not want to see where. We just want to know how much they are methylated. And then we quantified the quantity of immunoprecipitated messenger RNA. And the one you can look either here or on your paper, but in this figure A on the left, this is the quantification of M6A messenger RNA methylation. And we checked per 2 dBP until this is BR1. And as you can see, and we checked that at different times because we also wanted to know if there was any time-dependent methylation. But as you can see, for all per 2 dBP and BMR1, we have a reduction in the methylation at all time points. Okay? So all of them, the methylation of all these transcripts is inhibited by DAA. However, on the right of the panel A, this is the same thing, but we don't quantify M6A, we quantify the methylation of the cap, M7G methylation. And as you can see, deazoxyadenosine had no effect. So red is DAA, black is control, and as you can see, there is no inhibition here of methylation of the cap. How did we do the methylation of the cap? How did we quantify methylation of the cap? How could you quantify methylation of the cap? Now you know how to quantify methylation of mRNA, M6A. How do we quantify M7G? How could you quantify M7G? Yes. We use an antibody against M7G, and we immunoprecipitate mRNA with this M7G antibody. All right. So, now, this is nice. So, we show that M6A is inhibited, but not the cap, not methylation of the cap. So now maybe something nice is to show, okay, what is the effect methylation of inhibition of M6A methylation has on RNA? We want to know how it affects RNA. We know it affects the clock, but how it affects RNA. And at the same time we did this experiment, 2 papers got published, one in Nature, the other one in Cell, in which they reported that actually M6A affects the processing of RNA, how quickly RNA is degraded, how quickly RNA is translated. So we decided to check whether when we inhibit M6A, we have an effect on the processing of RNA. So this is the next experiment we did. And in the same figure, this one, as I told you, show the panel A show that M6A is inhibited but not M7G. So we decided to check whether, okay, when we use DAA, if we are inhibiting M6A, then we should see an effect on how quickly RNA is processed. And this is this figure here. This is a little bit difficult, but you can see this is a picture of cells. So this panel here and this one here, it's the same cell as this panel and this panel. So these cells are the same as these ones, and these cells are the same as these ones. These cells are the same as these ones, and these cells are the same as these ones. So this is the same thing. The experiment we did is at -2 hours. It's a little bit complicated. At -2 hours, so 2 hours before this picture, we added some sort of chemical to the cell that would label the RNA fluorescent. It would make the RNA fluorescent. If you look with a fluorescent microscope, you would see the RNA as bright red. Okay? So we labeled the RNA for 2 hours. After that, after 2 hours of RNA labeling, we removed the label. Okay, so RNA is no more new RNA will not be labeled anymore, but RNA has been labeled for 2 hours. So after 2 hours of labeling, we treat the cells with control or with DAA, and then every 2 hours, so here this is like 4 hours, we take a picture of the cells. The cells at 0 and the cells at 2 hours are not the same cells. They are different weights. Because we need, unfortunately, to kill the cell at each time point. So at 0, 2, 4, and 8 hours after the beginning of DAA treatment, we took a picture of the cells. And by fluorescent microscope, we can see that the cells that are not treated with DAA, very quickly they lose the fluorescence. At 8 hours, we cannot see anything. But the cells that were treated with DAA, as you can see, at 8 hours, we can still see they are fluorescent red. So what does it mean? What?
The mRNA was processed more slowly than DNA.
Yes, exactly. It means that the RNA is processed slowly. It disappears from the cell much more slowly. Okay, this is what it means. So when we have DAA, somehow, how quickly the cell gets rid of its RNA is completely different. Now, the blue one, do you have any guess of what it is? What is the blue one? Have you seen these kind of things before? Blue one is a very standard control, because here you see nothing. So people will think, how do I know that you have cells there? Right? So as a control, you label DNA. So you have a chemical that will bind to DNA and will make the nucleus completely blue. So this is what we have. So this is to show that at 0 and 8, both for control and DAA, we had cells there. And they had the same level of DNA. So they were the same, they were not sick or whatever. So this is what it shows. And of course, what you can do is you can quantify the intensity of the fluorescence from the nucleus, for example, and this is what you get. So at 0, there is no difference between control and DAA, but at 2 hours, there is a significant difference, and the intensity is higher in DAA, intensity stays higher at 4 hours, and intensity is still higher at 8 hours. So this shows that yes, RNA processing is slow under DAA treatment. And then the panel C, it's a little bit similar. The panel C, it's a very complex paper. Okay, the panel C, this is nice. This data is great, but it's not any particular RNA. It's just everything. Okay, it's just everything. What we wanted to show is 4 clock gene mRNA that are methylated, like BMAL1 and PER2. Do these messenger RNA also are they also processed slowly under DAA treatment? So this is what we wanted to know. Do we really have a difference in RNA processing of clock gene mRNA under DAA treatment? And then in this case, of course, you have control and DAA, control DAA, control DNA. On top here, this is BMAL1. At the bottom, this is PER2. And blue shows the amount of RNA in the cytoplasm. And brown is the amount of RNA in the nucleus. And we analyze cytoplasmic and nuclear messenger RNA after 0 hours of deoxyadenosine, 2 hours, and 4 hours. At 0 hours, there is no difference in the amount of nuclear RNA and no difference in the amount of cytoplasmic RNA. But at 2 hours, you can see that the fraction is completely different. The amount of nuclear BMAL1 RNA is higher under deoxyadenosine treatment, and the amount of cytoplasmic RNA is proportionally lower. And the same happens at 4 hours. The same for PER2, but the dynamic is a little bit different. It's not significant at 2 hours. It's not significant at 2 hours, but after 4 hours, the amount of nuclear PER2 is higher, and the amount of cytoplasmic PER2 is lower. What does that show? What does that show? What is one way of interpreting this data? Yes, that's right. This is what we wanted to say, is that processing is affected. It seems that the amount of RNA in the nucleus increased, but the amount of the cytoplasm decreased proportionally. So somehow the RNA remains in the nucleus. So nuclear export is inhibited by DA. Of course, because it's known that M6A is important for RNA processing and nuclear export. So we should expect to see RNA staying in the nucleus for longer. All right. This is linked to the previous figure. This is the second part of the same panel. Here, we also quantified the amount of pre-mRNA, pre-mature messenger RNA. What does it mean, pre-mRNA? Remind me, what does it mean? What does it mean, pre-mRNA? Yes, it's RNA that has not been processed. More precisely, RNA that has not been what? No? The introns, yes. So, pre-mRNA is RNA that has not been spliced before splicing. It still has intron and it still has exon. And of course, in this case, you can see that it's mostly brown. There is a very small here, very small blue thing here and here. But the amount of pre-mRNA was almost completely absent in the cytoplasm. This is, of course, as you expect, because only mature messenger RNA will leave the nucleus and go into the cytoplasm. So you would expect to have only pre-mRNA in the nucleus. And as you can see, what happens here is that after 2 hours, the amount of BMAL1 messenger RNA decreased dramatically. And the amount of PER2 also decreased dramatically. What does that show?
It shows that because the mRNA are exported out of the nucleus and remain in the nucleus increase and then led to the decrease of the transcription.
Yes. So you are right. So this actually shows that maybe not only RNA processing is affected, but also transcription. Somehow we are also decreasing the transcription of these genes. So maybe DAA does not only affect N6A or RNA processing, but has other effects, like decreasing the transcription. So we wanted to show this one as a control of what is happening. Okay? DAA seems to have other effects than only the processing. Or, as you said, because we affect the processing of RNA, this also affects the transcription. We didn't know here about the mechanism. All right. So the last panel, the panel D, shows that under DAA treatment, the stability of messenger RNA is affected, but not the stability of the protein. So this is very complicated again. On the left, you have actinomycin D, green, and actinomycin D plus DAA. And you have, in the middle, this is the same data but differently. In the case of the left part, we use Per2-Luc MEF. Do you remember what Per2-Luc MEF are? Do you remember what they are?
Proteins that is connected with the luciferase.
Yes, yes. So this is using cells that have a fusion between Per2 protein and luciferase. So of course, these cells express mRNA that is Per2 mRNA fused with luciferase mRNA. And so the protein is also fused. So just by looking at the amount of luminescence, you know what is the expression of Per2-Luc. So this is what we did here. But we actually treated the cells with actinomycin D. Actinomycin D, you may not know, it's an inhibitor of transcription. When we put actinomycin D in the cells cannot synthesize new messenger RNA, completely inhibited. So what the cells can only do is to make protein from RNA that is already there. Okay? And of course, the cell will progressively lose its RNA because each RNA has a different stability. Okay? So you can see from the beginning of actinomycin D treatment, the luminescence decreased very quickly. So this means, of course, that there is no new RNA being made, so therefore there is no new translation. And you can see that the cells that are not treated with DAA in the same time as actinomycin D, it sort of decreases a little bit faster than the cells that are treated together with actinomycin D and DAA. You can see the slope is a little bit sharper for only actinomycin D, which shows that maybe RNA seems to be somehow a bit more stable under DAA treatment. But this is not so convincing, so what we wanted to do is an experiment in which we show that when we use CHX, we don't have the same. So CHX, do you know what it is, CHX? Cyclohexamide. Cyclohexamide is an inhibitor not of transcription, it's an inhibitor of translation. So the cell can make new messenger RNA, but the cell will not be able to make new proteins. Okay? So in this case, when we measure luminescence, we only check at not the half-life of messenger RNA, we check the half-life of protein. Okay? And in this case, with or without DAA was exactly the same. So this means that the stability of Per2-Luc protein is not affected. Only the stability of the messenger RNA. But in addition, we checked the real amount of Per2 messenger RNA and we get the same data. So this shows actinomycin D without DAA, this shows actinomycin D plus DAA, and you can see indeed that at every time point there is still more HER2 messenger RNA when you put DAA. So it means that indeed the stability of the HER2 messenger RNA is increased with DAA, so that processing is faster. Okay. And I think I probably want to stop now and eat. What is next? The next is a bit more processing business. Yes, next is interesting, but I think that will be for next week. Do you have any question?
Yes. In the previous figure, we see that the transcription of HER2 decreased. So the transcription of HER2 decreased because the amount of mRNA, the HER2 mRNA is not exported to the it's not exported out of the nucleus due to the effect of DAA. In this figure D, we can see that it does not affect much. So I understand what you mean from this.
So you are thinking about figure C, the link between figure C and figure D, right?
Yes.
And you are saying that you are talking about this one here? Here?
Yes.
Okay, so you see that when you put DAA after 2 hours, you have much less pre-mRNA.
I mean that it is only affect the export of mRNA to the out of the nucleus, then it shouldn't have the difference between the relative luminescence of shouldn't be different if we inhibit the transcription. The level of mRNA shouldn't be different when we inhibit the transcription.
So you mean if we see a difference here, we should also see a difference there?
Then we should not show a difference there. In the previous, so what can we conclude from the figure C? Do we conclude that the DAA affect the export of the
Yes. Yes.
only affect the export of mRNA out of the nucleus, or does it affect the digestion or the degradation of the mRNA?
So this indicates that the nuclear export is inhibited by DAA.
Actually, that's one we guess from. That's just a guess, not one. It cannot be.
That's why I said indicate. I didn't say demonstrate.
So, what can we conclude from figure D, C, and D?
Figure C indicates that RNA processing is affected. In this case, nuclear export. And this shows that stability also appears to be affected. Of course, they are not mutually exclusive. They can be both. Maybe the stability is affected because nuclear export is affected. You see what I mean?
How can stability be affected by nuclear export?
That's a good question. What do you think?
Are there not as many proteins that break it down in the nucleus?
So RNA gets spliced and packaged with many proteins into the nucleus. Then, if the RNA has a good structure and a good sequence, it is exported in the cytoplasm. The mature messenger RNA is exported in the cytoplasm. And then, when it goes out of the nucleus, the proteins that interact with messenger RNA change. And the RNA interacts with cytoplasmic RNA binding protein. And there, the messenger RNA is degraded according to which protein binds to it. Okay? So, if Per2 mRNA is degraded when it goes into the cytoplasm, then if you keep it into the nucleus, it might also affect the degradation. You see? It's okay? But, okay, sometimes RNA is also degraded in the nucleus. There are many, many different things. We are not saying only this is happening, but we are saying, it looks like RNA processing is affected. We don't really say which mechanism, because there are so many of them that can be affected, but all these data together, so this means figure B, C, and D, shows that RNA processing is strongly affected by DA. And then maybe there are also supplementary figure that we added to these ones. And next week. Yeah, I'll explain that next week. You can read if you want, and if you have questions, don't hesitate. The most annoying thing for me when I did this thing was this. That was annoying. Because somehow there is a big decrease of the transcription. And I wanted this to not be significant. That would have been a more beautiful story. Because it would show that only the processing is affected, not the transcription. Regulation of transcription is something that has been shown many, many times. And of course, if you regulate the transcription of Per2, this can also affect the clock. So this is, you will see next week, is I explained this by saying that DAA is actually not that specific. And because it's a nucleotide, it can also affect directly transcription. Because the cell recognizes DAA as adenosine. And when adenosine is included into the messenger RNA, it stops transcription. So that's also not so good. So you will see next week. Okay, thank you very much. No, all right.